So at this point, we're familiar with the idea that the anomeric position, or carbon 1 of a monosaccharide, has the potential to act as an electrophile and undergo nucleophilic substitutions in which the anomeric hydroxyl acts as a leaving group. The example we looked at previously used an alcohol as the nucleophile to form an acetal. And one particular alcohol that's interesting to think about as a nucleophile in this reaction type is an alcohol group that shows up within a monosaccharide. So what happens when our nucleophile is itself a monosaccharide? Imagine, for example, that two monosaccharides were reacting. One was reacting through its anomeric carbon as an electrophile, and the other was reacting as a nucleophile through one of its other hydroxyls, say the 4-hydroxyl. In the presence of an acid catalyst, these two could condense with the loss of water in a nucleophilic substitution process to form a new acetal. And so where we had a hemiacetal in the sugar on the left, we now have an acetal, but that acetal involves a second molecule of monosaccharide in its structure. The nucleophilic oxygen at carbon 4 in the right-hand monosaccharide is here, and the electrophilic carbon, the anomeric carbon involved, is here, and the new bond was formed between the highlighted blue and red carbons. Because the product we've made contains two monosaccharide units, it's referred to as a disaccharide. And a disaccharide is formed most typically when an anomeric carbon in one sugar reacts as an electrophile and a different hydroxyl, potentially the anomeric hydroxyl, but often a different hydroxyl, acts as a nucleophile toward that carbon. And so this gets to the general point about why biomolecules contain both nucleophilic and electrophilic groups within their structures. This enables the formation of more complex structures like disaccharides from monosaccharides through self-reaction or reaction of the same type of molecule with itself. Repeated application of this reaction leads to polysaccharides, which we'll look at in a future video. The two carbons and oxygen atom that really form the core of the linkage between the monosaccharides and a disaccharide is referred to as the glycosidic linkage. And we distinguish between different types of glycosidic linkages using these numbers that correspond to the carbons that are involved. So, for example, in the hypothetical reaction that we've shown here, this is an example of a 1,4 glycosidic linkage, since carbon 1 in the left-hand sugar is involved in the glycosidic linkage, and carbon 4 in the right-hand sugar is involved. Let's look now at some specific examples of disaccharides. And it's a helpful exercise here to identify the glycosidic linkage and the numbers of the carbons involved so that we can give a name to the glycosidic linkage in each case. And let's start with sucrose, which is a very important disaccharide consisting of glucose and fructose units. The glucose is present as a pyranose and the fructose is present as a furanose. The glycosidic linkage involves the oxygen that bridges the two monosaccharides and the carbons that that oxygen is connected to. If we look at the monosaccharide structures individually, we can determine the numbers that correspond to these carbons involved in the linkage. In the glucose unit, the carbon involved is carbon 1, and we can see that this is the anomeric carbon because it's connected to two oxygens. It's part of an acetal functional group. On the fructose side, fructose is a ketose, and so carbon 1 is actually just connected to a hydroxyl group. Carbon 2, the ketone carbon, is involved in the glycosidic linkage in sucrose. And so one important thing to note about this glycosidic linkage that we're going to return to on the next slide is that both of the carbons involved in the glycosidic linkage are anomeric carbons. Carbon 2 in fructose and carbon 1 in glucose are both anomeric carbons. That has an important effect on the properties of this disaccharide relative to those of the other two. Let's move now to maltose, which is a disaccharide consisting of two glucose units. The glycosidic linkage in maltose is found here, and again, I think it's helpful to number the carbons involved in the glycosidic linkage. In this glucose unit, we can see that the carbon involved is part of an acetal, and therefore it is the anomeric carbon, or carbon 1. In the other glucose unit, we can determine the number of this carbon by looking for the anomeric carbon in this monosaccharide and then just numbering around. The anomeric carbon is here, it's involved in a hemiacetal in this structure. And if we number around, we see that carbon 4 is involved in the glycosidic linkage. And so in maltose, we have a 1,4 glycosidic linkage. Lactose is a disaccharide consisting of glucose and galactose units, and its glycosidic linkage is right here. In one monosaccharide unit, in the galactose unit, in fact, we find that the anomeric carbon is involved. Notice again, it's part of an acetal connected to 2 oxygens, and therefore it's carbon 1, or the anomeric carbon. In the glucose unit, we can do what we did in the case of maltose and look for the hemiacetal in this structure and start numbering the carbons from there. And when we do that, we realize that, again, carbon 4 is involved in this glycosidic linkage. So lactose is a 1,4 glycosidic linkage consisting of galactose and glucose units. Incidentally, just a little bit about these disaccharides. Sucrose you're probably familiar with as table sugar. It's a very common, very important form of sugar for nutrition. Maltose is a sugar that's important to the brewing process. It is one of the major sugars that shows up in unfermented beer, and so it's a key component of yeast food. It powers brewers' yeast as they convert maltose to glucose, and the glucose eventually to ethanol and carbon dioxide as part of the fermentation process. And lactose, of course, is an important sugar found in dairy products such as milk and cheese, and lactose intolerance relates to an inability to break down this disaccharide into its monosaccharide components.